So we already talked about the language of anatomy, all those terms you have to know. Do you have to know those forever and ever and ever? Yeah! Forever and ever and ever. Because remember, the rest of the book is written in that language. If you don't learn that language, say you didn't learn it very well for that test, it's going to be on your lab exam, which is this week. Again, go back and keep learning it. So if you made flashcards or however you did it, keep studying that stuff because you got to know that. Okay? We talked about the smallest building blocks of matter. What were those? Atoms, elements, organic molecules. What are they? Organic molecules. Organic molecules contain carbon. You are correct, sir. But organic molecules, give me some names. Molecules. Yeah? That's a hydrocarbon. Carbohydrates. Lipids. Proteins. Nucleic acids. Okay? Then we talked about cells. What makes up living organisms, or the smallest living unit. I can't get it to... I did the duplicate thing. I did all of the little things I was supposed to do. I'm plugged in. I don't know. Not showing. So we talked about cells, the smallest living units, living building blocks. So when I put the word living in, I want you to think of cells. And now let's talk about the cells and how they're all different. Okay? What'd you do?
I was trying to PC, but you want it for your laptop?
That's okay, I don't care. As long as it, because the laptop's projecting whatever's there.
I just hit, when it shows sources on the back.
That's okay. Yeah.
I hit that again. You may have done it, maybe it just...
Yeah, I did that, and then I did you can press on the keyboard, you can do the Windows P and have it do, and then that didn't work. See, it just likes you. It happens every time.
We had to scare it, I guess.
Yes. Put your foot down. Thank you, hon.
You're welcome.
All right, that's going to come up on the recording, by the way. That's all right. You know what I'm saying. And we didn't say anything bad. All right, so living unit cells. Now let's talk about how those cells come together to form tissues. So, tissues. What's a tissue? Groups of cells, groups of similar cells. And groups of similar cells will come together to create some of the very familiar organs that we see and organ systems. So we're moving on as far as arrangement goes in our discussion. So these are some of the 4 basic types of tissues we see in the different systems we're going to talk about in this class. It's very important, if I'm going to understand the system, that I understand the cells and how they work at the tissue level. So in an anatomy and physiology lab, you're going to do a lot of microscope work. You're going to look at those cells and those tissues, and you're going to study them in more detail. So, in order to look at these cells, I have to prepare them for viewing. I have to make sure, because it's a compound light microscope, that they're thin enough for what? Light to go through. And because these cells naturally are clear, what else do I have to do to them? I have to add some color so I can see them. You did that when you did your epithelial cheek cells. Did everybody do that in lab? Who did not do that in lab? Because you missed lab, or you're not taking lab? Did you ever do it? Okay, remember you put the stain on there? So you could see the cells, right? Okay. She says yeah. She's like, no, but I'll just say yeah to shut her up. In order to see them, the way we see them in a lot of the pictures that you're going to look at in these textbooks in your lab manual, you have to add color to them. You have to add stain to them so you can see them. So staining procedures are extremely important when we want to actually view these groups of cells or tissues. The first group of cells that we're going to talk about form a tissue called epithelial tissue. And epithelial tissue's all over the place. It covers things. It lines things when we talk about some of the different organ systems in the body. The whole outside of your body is what? Epithelial tissue. The lining of all your arteries and veins and the inside of your heart, what is it? Epithelial tissue. So it's extremely abundant, and we're going to find it throughout the entire human body. Some of the special characteristics of epithelial is it is what we call polarized, or it has a polarity. What was a polar molecule when we talked chemistry? It's something that had opposite charges, slightly positive, slightly negative end. Well, these sort of have opposite sides when we talk about epithelial tissue. When we look at epithelial tissue, the apical surface is something on the outside. So if it's lining a blood vessel, it's what's touching the blood that flows through. The basal surface sticks that epithelial tissue to something else. So that's the underneath side, let's call it. There's a bunch of proteins there that are produced by a layer underneath, which is similar to connective tissue, along with the epithelial cells to create kind of glue that holds the epithelial tissue to what's underneath. That's called the basal surface.
That's like you fall down and you get like a skin tear.
And it's not bleeding?
Right.
Yep.
Yeah.
You've ripped away the epithelial, but underneath is connective tissue. The other very important characteristic, so glad you brought that up, of epithelial tissue is it's avascular. It needs blood, but it doesn't have blood vessels running through it. So without blood vessels. Where do the cells get the nutrients that they need to survive in epithelial tissue? Diffusion of nutrients from underneath. So that basal surface creates a layer called the basement membrane. And that is a surface that allows nutrients to come from those blood vessels. And we're going to find out they're underneath in a layer typically called connective tissue. So epithelial tissue is connected to connective tissue via the basement membrane. So the basal surface is kind of a sticky end, and that's where it connects to the connective tissue. Another thing we're going to find lots and lots of in epithelial tissue is tight junctions and desmosomes. You know what those are, right? Those are connections. What kind of connections? Like really, really tight ones, exactly, like spot welds. Remember we talked about desmosomes and we said they were kind of like a spot weld? That's kind of good, isn't it? Because I don't want to do this and have my entire integumentary system rip off, right? Kind of nice if it was held on there pretty well. Actually, when I do this, and this visual is getting kind of old. Does everybody remember Charlie Brown? Who doesn't remember Charlie Brown? Whew. I can still use the material. Remember Pigpen? When he walked along, it looked like dust clouds were coming off of him. Well, if you had a microscope to follow you along, you're Pigpen. Because that outer layer of dead epithelial cells falls off of you as you walk along. It's disgusting, isn't it? Dust in your house? Yeah, you know what it's made of? Your epithelials. Yeah. No, it'd be even grosser. It's your epithelials and a bunch of dust mites that eat your epithelials. Your mattress, when you buy it at the store, give it 5 years. It weighs a lot more. How come? Your epithelials. Yep. So, outer layers of epithelial may sometimes be what? Dead. This? Dead. Why is it dead? Remember, epithelial tissue is avascular. The further away it is from food source, it's going to die. So as we go and we look at the classification of these tissues, we're going to see that we have a group of epithelial called stratified. That means more than one layer. And that's what your outer layer is. It's stratified squamous epithelial tissue. So the very outer part is way far away from the food source. And that's where my little scrape analogy comes in. So if I scrape my surface and just scrape off the epithelial part of my integumentary system, and we're going to discuss later on in the semester that the integumentary system is made up of two different distinct types of tissue. The epithelial layer on the top, that avascular stuff, and underneath, connective tissue. We call the connective tissue dermis. We call the epithelial layer what? Epidermis. So if I just scrape off that top stuff, am I going to bleed? Nope. I have to break into what? Yeah, the underneath, the connective tissue part, and that's when you'll bleed. That's where the blood vessels are. Okay? So, they're very highly stuck together. And specialized contacts like desmosomes can be found throughout. Underneath, we have some sort of connective tissue that it's connected to. It creates something called the basement membrane along with parts of the connective tissue underneath called the reticular lamina. So, it's avascular. And if I'm constantly sloughing off cells as I walk, do you think I might need to make more? Yeah. Epithelial tissue has the ability to undergo mitosis at a very rapid rate. So your cells in epithelial tissue are constantly regenerating themselves. One of the targets for chemotherapy drugs are cells that reproduce themselves at a rapid rate. Does anybody know, or has anybody actually experienced the effects of chemotherapy? What's one of the things that might happen? Lose hair. Do those cells, which happen to be epithelial cells, regenerate themselves at a fast rate? Yes. Yeah? How about these? Yep. Another complication might be problems with fingernails. You might actually lose them. Because the chemotherapy drug doesn't know the difference between your normal epithelial cells and that abnormal group of cells that it's targeting to kill off. So that's one of the complications. You start to lose cells that reproduce themselves at a fast rate, even the normal ones. So epithelial tissue has the ability to regenerate itself at a very fast rate. So now we have to learn some terminology. How do we classify these tissues? We classify them based on shape and arrangement. If I have a single layer of cells, what kind of tissue is that? Simple. If I have more than one layer, stratified. If the cells look like floor tiles, flat, squamous. So I can have simple squamous, or I can have stratified squamous. And we always throw the word epithelium on the end. So see now where those big words come from? If I have kind of chubby cube-shaped cells, I call them cuboidal. I can have simple cuboidal, single layer of cube-shaped cells with basement membrane and connective tissue underneath. Or I can have stratified cuboidal cells attached to basement membrane with connective tissue underneath. So every time I describe epithelial tissue, don't forget to talk about the basement membrane and the connective tissue underneath. And last but not least, we have something called a columnar shape or column-shaped cell. Simple, stratified. Yes? There's also another little delineation in there called pseudostratified. What's pseudo mean? False or fake? So these cells are actually a single layer of cells, but the way they look under the microscope makes them seem like they're stratified. We'll call them pseudostratified. So we'll introduce you to those in a minute. In this chapter, what you need to understand is how to describe epithelial tissues. Yes? Simple, stratified, shape, squamous, cuboidal, columnar. And then you have to know a little bit about where you might find these tissues. Now these are nice because they give us two representations of the tissues. Over here, under the term simple squamous epithelium, you see an illustration of what they kind of look like. And then over here, you see some actual histology of that particular tissue. The other thing you find in these nice little diagrams is where I might find this type of tissue. Kidney of the glomeruli, in the glomeruli of the kidney, excuse me, air sacs of the lungs, lining of the heart, blood vessels, etc. The other thing, what it's for. So this is simple squamous epithelium. A single layer of what? Flat, tile-like cells with basement membrane, connective tissue. Don't forget, that's important. This is a photomicrograph of simple squamous epithelium that line the alveoli. Does anybody know what an alveoli is? It's like a balloon inside your lungs. When you breathe in, air comes down these tubes and fills up the balloons. The balloons, here's the inside of a balloon. Here's the inside of a balloon. Here's the inside. You see that? These cells, simple squamous epithelial cells, are here. But watch this. This is what the cell looks like. Yes? That's what you're looking at. Does that make sense? So I'm taking the balloons and I'm bringing over a sample of tissue to an instrument called a cryostat. You don't have to write this down. I'm just explaining the way it's made because maybe it'll make sense. So I take the tissue, remember the three-dimensional tissue, the alveoli, and I bring them over to this thing called a cryostat, which is like a salami slicer on steroids. Okay? And see what you're looking at now? So these are the cells. In this form, we'll see them different ways looking at different tissue. So simple squamous epithelial cells. Understand where I might find them. Understand what they're for. A simple layer of epithelium is only going to require one layer of cells to pass through. Right? Yeah, so let me go back. Where might I find simple layers? In places where I can have things pass from one place to another. So in the alveoli, what has to pass from one place to another? Respiratory gases, right? Blood vessels. And they probably were here at one time, or running alongside these cells. So from the sac can pass oxygen into the circulatory system. And watch how fast it happens. Ready? Done. Yes? And then waste gases, carbon dioxide, can pass from the circulatory system into the sac so I can get rid of them. Yes? So simple layers are in places where exchange needs to happen. We need to have a separation, but we need to have exchange. Cuboidal-shaped, simple cuboidal epithelium are cube-shaped epithelial tissue with basement membrane and connective tissue. Here, we need the cells to kind of do a little bit more work besides the quick that the simple cells did. They might have to grab things and push things out the other end. What's that called in science term stuff? Grab things from the outside of the cell. Endocytosis, exocytosis, does that ring a bell? Phagocytosis is an example. Pinocytosis is an example of endocytosis. Is this all ringing a bell? Oh yeah, shoot, you had to remember that. That's where we're going to find those cube-shaped cells. Places like the kidneys. What do the kidneys do for a living? They filter filter what's in your circulatory system. They filter blood plasma. They get rid of waste products for you and excess water and excess hydrogen ions. They help with water balance. So the cells that are here, see them? How many cells do I have in this little tube lining this tube? 1, 2, 3, 4, 5. Remember, salami slicer. So what if I take this and the cells lining it, this, and I make a cut through it, and I cut off the nucleus of this cell and that cell? Yes? So remember, you're looking at something three-dimensional that's been cut on a cryostat.
So how do you tell that between like that?
Because I'm wicked awesome.
Sure.
Well, see these?
Yeah. Okay.
You can actually see the plasma membrane of each of those cells. So here and here and here and there. It's hard to see up here because it kind of gets blurred. If you look in your book, you can actually see it a little bit better. This one's a better example. You can actually see. So see the plasma membrane of this, and then this, and then this. Okay? So these are simple cuboidal lining tubes. What are they for? Secretion and absorption. So endocytosis, exocytosis stuff, where I have to grab from one side and push out another side. Kidneys is a good example. Ovaries, glands that secrete hormones. We're going to find this type of tissue. We good? Yes?
What were those, say those 5 cells together, what were they called when they're all together?
They're called simple. Simple what? Simple cuboidal epithelium. Sure, if you want to throw that in.
But the dark pink stuff, is that the connection?
Under here, see this dark pink line?
What's that big?
That's basement membrane.
Right, what's the big dark pink stuff?
This?
Yeah.
That's a blood vessel. So here's a blood vessel right here. Those are actually red blood cells. It's kind of cool. You see it here too. This is a nice side section of a blood vessel. So these are all red blood cells.
In our lab, I think you put the center part of the surgical one, some kind of something like that.
It's a hole, right?
But she called it a hub of some kind.
This?
Is there any significance to the video?
This is a lumen. It's a space. It's a hole.
Okay, that's some difference.
Well, it's a space where stuff passes through. So which is the apical surface? This end or that end? This end near the what? The hole, or the, give me the science name, lumen. Yes? This is the basal surface. Where the what's underneath? See the dark pink line? Basement membrane, that's the glue. All of this stuff in between is connective tissue. This is a blood vessel. So when you go to look at your tissues under the microscope in lab, you have to remember you're not looking at just simple epithelial cells. You're looking at a piece of tissue. So where do you think you're going to find the epithelial layer on that piece of tissue? In the middle?
You mean the apical or apical?
The epithelial layer. Where do you think you're going to find it on a piece of tissue? It's going to be either here on the top or on the bottom, depending on how they put the piece of tissue on your slide. So I'm just giving you a little hint. When you go to look for epithelial tissue, it's going to be covering or lining something. The next, simple columnar epithelial, a single layer of column-shaped cells with basement membrane and connective tissue. Again, thicker cells, we're going to see more exocytosis, endocytosis function. Thicker cells are also going to give me a little bit more protection between what's on one side and what's on the other side of these epithelial cells. So we see, again, absorption and secretion in the description. Another thing we might also see in this type of layer is extra little cells that are sandwiched in between the cells called goblet cells. And they produce mucus. So in places where things need to slide along, need a little help, we might also find sandwiched in that layer these mucus-producing cells. Places like the intestines, the digestive system, some of our reproductive organs, uterine tubes are places where I might find this type of epithelial tissue. So on this particular micrograph, I have a couple of extra little parts. I have the cells, see them here? They look like big tall buildings. What's this? The nucleus. So this is nice. We have all the nuclei all lined up like little soldiers. What's this? Basement membrane. It's better defined right there. And what's underneath? Connective tissue. These are the goblet cells. And they kind of look like crackled stained glass under the microscope. Yes?
Now, is the goblet cells, is that what dark reticulated is, or that's
No.
that's the
It's called hostra. And they're little pockets inside the large intestine. And sometimes things get stuck in them.
right. That's when they're going to take them to tools and
Yep.
step along.
It's the reason fiber is your friend.
It's not, they don't, yes,
Yes.
they don't occur in them.
Well, what is this?
Wouldn't that, right, but the top part's the intestine, correct?
The top part is the tube.
Right. So
This is a small intestine, though. So you would see that in the large intestine. The other thing we're going to find here is an extension on this particular layer that is going to allow for increased surface area. So if I have a surface area this big, and I can make it this big, I can do a lot more work at the surface, right? If I take these column-shaped cells and now take the edge of them that's exposed to what's inside the lumen and create these little extensions. I can take this much area and bring it down to this much space. Does that make sense? See what I did? So if I had a string that I could line across my fingers like this, and I wrapped it like that and then pulled it apart, I would see it would be about that long, right? Microvilli on these cells is going to increase surface area. What goes on in the small intestine, do you know? Absorption of all the goodies that you put in your mouth, yes? So those cells from that lumen or space have to suck out all the goodies and pass it down where? To the connective tissue, into the circulatory system for transport. So that's where we find these guys. Yes? Yes, sir. It's on. Okay? So this is where we find this type of cell. Yes?
So are they like making that mucus, or is that just...
These guys are. These guys are making mucus, and where are they putting it?
They're putting it into the small intestine.
Out onto the surface of that cell layer, out into the small intestine, the lumen. Yep, different cell. This is not an epithelial cell, it's a goblet cell. And it hangs out in the epithelial layer. The mucus is going to help things move along the tube. So think about what you put in your mouth. That's going to go into your small intestine, right? It's going to move through that tube. If it gets caught up in the tube, what's going to happen? You're not going to be very happy, right? Things get backed up. They don't move along. You can't absorb properly. It'll stop the works. This is the example of that other type of stratified I was talking about. This is called a pseudostratified layer. It's actually a simple layer of columnar cells, but they're not column-like. They kind of get skinny. So here's an example of a cell, this one. It actually attaches down here, gets chubby here, and gets skinny up here. Did you see that? So here it gets skinny and attaches here, gets chubby and goes to the surface. This one will do the same. Get skinny and attach here. So this pseudo-stratified delineation is found in these columnar cells, yeah. We don't typically see it in other ones. No, we'll see something else. We'll see something else called transitional epithelium. All right, these guys are found in the trachea, lining the trachea. They also have an extra little part on the top. Kind of looks like a crew cut, yes? That's called cilia. A little bit more than microvilli and more hair-like. So these cells are pseudo-stratified, a single layer of column-shaped cells that appear to be stratified with basement membrane and connective tissue. So we find this type of cell in places that we need to move things across that cell layer. Trachea. What are we going to move? Mucus. What's the mucus for? When you breathe in, everyone breathes in perfectly sterile and filtered air, right? No, there's a whole bunch of schmutz in the air. Do you want that stuff down in your alveoli? No, no, no. So as you breathe in, the schmutz gets stuck in the mucus. Yes? So it's kind of a filtering mechanism. You have a bunch of it in your nasal passage and down your trachea. So that hopefully by the time air gets down into the alveoli, it will be schmutz-free. So where does all the schmutz go? It's stuck in the mucus. Yeah, what's the cilia doing? It moves the mucus back to a much safer region in your body. Guess where? Into your mouth, where you're going to do what with it? Swallow it all day long. You find yourself doing that every once in a while, swallowing? Yeah, you're swallowing the mucus with all the schmutz in it that was swept up from your trachea. Now, got a little disgusting talk for you. Yes?
Is this layer more bendable because...
The cilia is.
But I was wondering if the others...
No.
They don't.
No, they're not, the cilia is. Okay, so these are, ready for the word? Ciliated, pseudostratified, columnar epithelial cells. Whew. So the cilia creates wave-like motions that are going to move the mucus across the surface of this group of cells. Who smokes? Raise your hand. If you smoke, raise your hand. Be proud of your cigarette habit. Go ahead, show us. See, a lot of you are like, I'm not raising my hand. She's going to insult me. Yes, I am. Because when you smoke, you breathe in really schmutzy air. Yes? And where does all this stuff go? Gets stuck to your mucus. But the smoke also does something to your cilia. What's it do? It paralyzes it. Every time you smoke a cigarette, for about 20 minutes, your cilia are paralyzed. They can't move that mucus. So where does the mucus go? Yes, ma'am. Yeah, that's part of the talk. Wait for it. So, yeah, I love it. So, you paralyze your cilia about 20 minutes every time, and that it stays, it's in order. So 2 cigarettes, 40 minutes. 3 cigarettes, 60 minutes. You can do the math, right? So you smoke all day. You're still breathing in air with schmutz in it. It's still getting stuck in the mucus. What's it doing? It's sitting on all of those cells, festering throughout the day. So you finally stop smoking at the end of the day. Hopefully you don't smoke in your sleep. You rest, you lay your head down and go to slumber. And in the middle of the night, at some point, what happens to your cilia? It starts to wake up. So what's it going to start doing? Waving all that yucky mucus that's now been festering with all the schmutz for the entire day into your mouth. So you wake up in the morning and you start your day. Let's go have a cigarette. Yes? Okay? So not only do you paralyze cilia, you destroy these cells. You destroy these cells. Anytime you aggravate cells, what can you do, or what might happen to them eventually? They can cause mutations. Mutations can lead to cancers. How much do cigarettes cost? Those of you who smoke, give me a number. How much? You can roll them? Holy moly. I always love the rolling paper people. I roll my own. It's very cheap. Yes, you kill yourself cheaply. That's nice. So, yeah. So about, I don't know, $7 a pack? Yeah. How many packs a year? You do the math. Stop smoking, save your money, and take me to the Bahamas. All right? As a thank you for giving you the disgusting smoking talk in class. All right? So, cilia, very important. Ciliated cells in the trachea are the pseudostratified kind. Yes? Now we're talking about what kind of layers. Those were all the simple. Now we're talking about what kind of layer? Stratified, which means what? More than one layer of cells. The first one they talk about is stratified squamous epithelial cells. Many layers of what? Flattened. With a basement membrane and connective tissue. So now we have many, many layers. And when we start to see many, many layers, stratified layers, protection is usually a word that gets thrown into the mix. So this, on the outside, is protection. It's stratified squamous epithelial cells. This stuff also has an added extra little chemical in it called keratin. Does anybody know what keratin does for me? Yep. It's a waterproofer. So you're also going to find some waterproofing in those outer stratified squamous epithelial layers. So when you go in the water, you don't blow up like the Stay Puft Marshmallow Man, right? Because water is what kind of solution? What kind of solution is water? It's a hypotonic solution. You need a little waterproofing surface there. Okay? We also have stratified layers in places like the esophagus. And this one is a micrograph of the esophagus. Now remember, cells, basement membrane, connective tissue. Connective tissue is my food source. So in this particular micrograph, the connective tissue is under here. The basement membrane is right around there. And these are the cells. The live ones, can you see them where they are? Where are the live ones in this layer? They're closest to the food. So it's kind of like, you know that song, did you ever sing that song, Little Bunnies in the Bed song, when you were little? Like one jumps in and everybody rolls over and the other one falls off the end song. Bunnies or monkeys. You didn't sing that song? Roll over. I'm not going to sing it. Right? So one rolls over and the other one falls out and then 7 in the bed and the little one said, yeah? No? Yes. Okay, good. Think of that song when you think of how epithelial tissue grows. So the living cells, and when we talk about the integumentary system, we'll name all these layers. But the living cells are going to be closest to the food source, connective tissue. They're going to grow, and as they mature, where are they going to go? They're going to roll over in the bed. They're going to move up towards the what end? What end? What end? Apical end. So they grow at the basal end, and they move towards the apical end as they mature. They're going to hit a certain point where they're going to be too far away from food to get any. So what's going to start to happen to them here? They're going to start to die. So those outer layers are dead, but they still provide protection for that tube. In this case, the esophagus. Think back to breakfast this morning if you chose to eat it. Anybody eat breakfast this morning? See, I asked the opposite question. Who ate breakfast this morning? Very good. You chew your food. You swallow it. Your esophagus is about that big. Is that about how big the food was that you swallowed? Most likely not. So what has to happen to your esophagus in order for you to swallow your food? It stretches. What happens to the food as it goes down? It tears along the sides of the esophagus. Yes? Pulling with it what? That outer dead layer. So every time you swallow a little bit of yourself. That's another disgusting word. Yeah, we're going to throw all kinds of disgusting ditties at you in this class. But as you swallow, you're pulling off those cells. Do you see why you need this kind of layer in places like the esophagus? Because if you had a simple layer of cells, you'd get one swallow a day and that's it. You'd be done. Because you would pull off those cells, that wouldn't be very efficient, right? So stratified layers provide protection to tissues that they are lining or covering. Where do we,
When the cells are created in the stratified, are they generated like basement level up?
Yep.
Or
So they generated in the basement level, and then they move this way. So the next one in line that's generated pushes the already cell there. So everybody keeps pushing that way, just like the bunnies in the bit. Yes? Until you get so far away, you're going to start to die, and then eventually you're going to fall off. So we have two different types. We have keratinized and non-keratinized. Waterproofed and non-waterproofed. The keratinized ones, all over the outside. Non-keratinized, lining important tubes, like the esophagus. The other type of stratified epithelium we're also going to see in our bodies is something called transitional epithelium. These guys are in places that stretch and expand. They give us an example of the lining of the uterus or the lining of the bladder. These are in the bladder. What's the bladder do for a living? And some of you might need to go right now. Yeah, it's a holding tank. Your kidneys are constantly filtering your blood plasma, getting rid of waste products and extra water and extra hydrogen ions. Constantly. If you didn't have a holding tank, what would you be doing all day? You would be peeing all day. Okay? Not efficient. So you have a holding tank which fills up, and then you'll get a little message that says, tank's full, and then you have to go empty. Yes? So in places that stretch like this, we're going to find something called transitional epithelium. Very similar to what we just talked about, except what we're going to see is not tons and tons of layers, but cells that change shape within the layer. So at the bottom where they're born in the basal layer, they're kind of small. And by the time they reach that outer layer, what happens to them? They get big and bubbly. Okay? They transition in shape in transitional epithelium. So places that stretch and store things, like the bladder, we'll find transitional epithelium. Is transitional stratified or simple? Stratified. Yes? Excuse me. So, the next thing the book talks about is glandular material. What's a gland? Epithelial tissue glandular tissue is epithelial in nature. What does a gland do for a living? Secretes something, like what? Hormones. So the cell makes something, packages it, and ships it. Who helps me do that? The Golgi apparatus. So, when we look at glandular epithelium, we see cells that have tons and tons of Golgi apparatus and these little secretory vesicles that help deliver the product that they're trying to deliver. We see that the cells that make this product, whatever it is, can be arranged in many different ways. The two major types of glandular material that we're going to talk about in this class are ones that take the product that they make, squeeze them out of tube to somewhere else, or ones that take the product they make and just spit them right outside themselves into the circulatory system. So what's the difference between the two? The one that spits its product out right outside itself into the circulatory system, what kind of gland would that be? Endocrine gland. There's a whole chapter, second semester on it. The endocrine system. The other type of gland, also epithelial in nature, is called an exocrine gland. Example of an exocrine gland. Yeah, tears, lacrimal glands, mucus, some mucus producing. Okay, sweat. That's an example of an exocrine gland. So the cells that line these glands make the product and squeeze them out of tubes somewhere else. So we can have simple duct structures where we see the cells lining something and squeezing stuff out. We can have branched, and we see this down in the stomach region, producing gastric juices for you so you can digest your food. We can have compound glands. In the small intestine, digestion takes place, lots of absorption takes place, but one of the things that's going to help me digest my food, break it from large to small, is enzymes. So these guys, in the beginning portions of the small intestine, are going to squirt in lots and lots of wonderful enzymes for that process to take place. Then we can also have simple and branched alveolar. Sometimes we don't see all of these in humans. Might see them in dogs, cats. Scent glands. Anybody hunt?
Oh, yeah, that's terrible.
Yeah, they're nasty. Okay. But what are they for? What are sense glands for in the animal world? In the other animal world? You do it too, by the way. Yeah, to sense signals. We, as humans, aren't very good at sense signals. Not like a deer or a moose or any of the other animals in the animal kingdom. They have a very highly developed olfactory bulb, and they can pick up sense signals much better than we can. We kind of have dinky, inadequate olfactory bulbs, not compared to a dog or a deer. So we'll see those in some other organisms. Sebaceous glands that produce oils. On top of that layer of all that dry, dead skin, you're producing oils. The oils are there to help with the waterproofing thing, but what else? What happens to you when your skin gets all dried out? It starts to crack and it starts to peel. Is it going to be very good protection for you if it does that? So you have sebaceous glands in your integumentary system that produce oils to help soften that up so we don't get cracking and splitting. We have compound alveolar glands called mammary glands. And what do those guys produce? Milk. And then salivary glands. What are salivary glands for? Spit. What's spit for? Exactly. When I start the digestive process, it's important for me to break big things into small and add some moisture, add some ions, add some enzymes to start the chemical breakdown. And that's what saliva is going to do for me. So these are some examples of these glands. So we have multicellular exocrine glands. We have things called miracrine glands. And what do those glands do for you? Not saying that word today. How do they secrete their product? Exocytosis. So they make it inside the cell and they package it up and they ship it. Yes? What's a holocrine gland? So they don't all do it the same way. Yeah, martyrdom, exactly. They explode. So they accumulate their product within them until they explode and release their product. And again, the way the cells are set up, the new ones down here being born all the time, down at the basal surface, pushing their way up to the surface when they explode and release their product.
Where are those
Places like your, think of the stinky secretions.
Oh, yeah.
Yeah. Yep. Yeah, again, these are epithelial cells. They're just glandular in nature. We also have apocrine glands. And again, according to your textbook, and I quote, are present in other animals. There is some controversy over whether humans have this third gland type. So we're not sure if we have them or not. There's always an argument as to whether a gland is apocrine or holocrine. Not sure. But again, because epithelial tissue constantly is reproducing itself, we are constantly regenerating these cell surfaces. So those are epithelial cells. They can be simple, they can be stratified. They can be squamous, they can be cuboidal, they can be columnar, they can be pseudostratified, they can be transitional. They're always going to be cells, avascular, with basement membrane and connective tissue. Okay? So know them all. Be able to identify them visually, because guess what you're going to do on your next lab exam? Yep, there's going to be a whole bunch of microscope work to do that on that lab exam. So we're going to have to be able to identify them visually, not only from pictures, but from slides. The next classification of tissues is connective tissue. Where am I going to find connective tissue? Under the basement membrane. Very good. Oh, you are good. Oh, I'm not done. Oh, shit. Shoot. You didn't hear that. Sure you did. It's on the recorder. Okay, there we go. So, oh, shoot. Shoot again. I forgot to start the clicker program. Get your clickers out. Well, I'm figuring out the stupid computer. All right, you ready? All right. So if I'm going to prepare a microscope slide, put the following steps in the correct sequence for preparing a specimen for microscopy. Yep. Remember my exciting salami slicer on steroids discussion? Five, 4, 3, 2. There's only 50 people today? 51? What's the answer? The answer is B. Okay, let's go over this again. So no, I didn't give you these terms. So, when I want to look at a piece of tissue, say I get a sample from the operating room, somebody got their gallbladder out or somebody removed something. What I have to do is create a specimen that can go to the salami slicer that I can cut. I have to put it in wax. It's called paraffin wax, and I freeze it. And then I bring it to the salami slicer. And then I'm going to section it. So I'm going to make slices. Okay? The slices are really, really, really thin. So then I have to take those slices of tissue and I have to lay them on that glass slide. Because I'm going to need that to look under the microscope, right? In order for me to then put the stain on, I have to make sure that salami slice that I made sticks to the slide. How do I do that? I have to fix it. That's wrong. It's section, fix, stain. Yeah, that's wrong. So, so I have to fix it to the slide. I have to stick it to the slide. You can do that chemically or heat fix. Okay, but you fix it. Hold on 1 minute. Let me finish my explanation. So you stick it to the slide. Once you've stuck it to the slide or fixed it, then you can apply the stain. If you don't do that, it's going to fall off. Question? Fixing is also to the slide, too. So I would never ask you that question. That's stupid. It's a book thing. It's a book thing. Yes. In my brain it is. But the book says fixed in sections and stain. But again, you have to create or get the tissue, put it in paraffin, make a slice, fix it to the slide, and then stain it. In my mind, it's A. Which of the following relationship between a tissue and its general function is not correct? Five, 4, 3, 2, 1. What's the answer? A is the answer. Nice. What's epithelial tissue for? Protection. And one more, and then we'll go back to connective tissue. An area of the body exposed to the external environment would be composed of which of the following tissue types? Five, 4, 3, 2, 1. What's the answer? A. Who's got their muscles all hanging out? Anybody have their muscles all hanging out? I know you work out, but they're covered with something, right? Caesar. Okay. Oh, wait a minute. All right, let's go back. Connective tissue is next. So connective tissue, I can't see this damn. Connective tissue is found where? Under epithelial tissue, right? Connecting the epithelial tissue to something else.
I have a question.
Yes, dear.
There was a question on the master about cartilage and bone.
Sir.
Pertaining to, it says that it is firm and hard. And then in the book, in one of the little graph, in the figures. It says that it's a gel-like substance. Well, how can it be hard and firm if it's a gel-like substance?
Which is it? What are you asking me?
Cartilage and bone tissue.
Cartilage?
As one of the answer was that the bone tissue is hard and firm. But
Okay.
in the book it's saying that it's jelly-like.
Your bone is jelly-like?
That's what it says right here.
It says the matrix of it.
The matrix of the bone is jelly-like. Think of a bone.
Yeah, I know. I guess
Again, what was the question? So, and what answers did it give you? If it said firm and hard and it gave you something like blood or epithelial
No, that was
as a choice.
No, there was cartilage and bone tissue. Maybe it was both have what? And then one of the answers was, let me see, that they're something hard. I don't know.
Yeah, both have, and we're going to talk about that right now. All connective tissue has very similar parts. Now, what's an example of connective tissue? Softer stuff that's underneath your epithelium. But what's also connective tissue? Bone. What's also connective tissue? Blood. All of these tissues, connective tissue, connecting one thing to another, have similar parts. This diagram shows us some of those similar parts, some of those similar structures that we're going to find in all connective tissues. But we're going to find them in different amounts, which will give the connective tissue different structure.
That's what I'm saying. I mean, like,
That's why I don't assign those. Didn't we have this discussion? Those aren't my questions. So when you run into one of those stupid questions, don't get caught up with it because...
Yeah, but it affects your grade and it's wrong.
No. It doesn't affect your grade. It's not a lab question. It's a lecture question, isn't it?
No, this is a lab question.
Oh, all right. Then yell at your lab teacher about that.
Yeah, but I'm just saying.
No, I understand. And some of them might be that way. And you should bring that up in lab.
Yeah, I will.
And I can't, I have to actually look at the question to answer your question. Yeah. So it's hard to do that. Yes, hon.
What's your name again?
Michael.
I noticed on the table 4.1 on page 129, it says that they're gel-like substances, but that cartilage resists compression, but that bone tissue is hard tissue.
Right. And it's because of what I'm going to explain in a second. That all connective tissue has these parts, but some of them have more than others, which is going to make the connective tissue that we're talking about more dense or less dense. So,
Oh, I was just telling you.
No, I know. And again, if you have a question about mastering questions, bring it up in lab. You guys can discuss it, and then you can teacher can decide whether to pitch that question or not.
I was just asking.
No, only because
Jelly-like, but the
connective tissue is jelly-like in some forms, yes. Your bone comes from jelly-like material. When we talk about bone production in the bone chapter, you're going to see your bones came from cartilage. So at one point, they were jelly-like. So it depends on how that question was asked.
yeah. I just wanted to try to
Yep. So at one point, they were. And it's because of the substances that connective tissue are made of. So what is connective tissue made of? Well, all connective tissue has some sort of organic material that makes up what we call the matrix of that connective tissue. So all connective tissue has ground substances in it that forms sort of the background that all these cells sit in. If you noticed in the micrographs, the connective tissue was not all cells lined up one after another, like epithelial tissue was. Connective tissue cells, when we find them in connective tissue, tend to be much more spread out in this matrix. So when we're talking about cartilage, which is connective tissue, versus bone, which is connective tissue, versus blood, which is connective tissue. All of them have ground substances that make up those matrices in that tissue, but it's going to be in a different form. So in blood, what form is the matrix in? Liquid form. In cartilage, we're going to have more substances which start to make it firmer and tighter. In bone, we're going to have, we're going to see calcium and phosphorus and magnesium salts that actually make the matrix hard. So which of those tissues has more ground substances in it? Bone or blood? Make sense? So the amount of those substances and the form of those substances are going to determine what kind of matrix we have. So it's going to differ between connective tissues. The other thing we're going to have in connective tissues are what we call connective tissue fibers. And what do fibers do in any material? They're going to add strength. Again, depending on what we're talking about, we're going to see those fibers in different forms. We see fibers that are very thick, almost like telephone poles, that are going to add a lot of strength to some of our connective tissue, called collagen fibers. We see that they're not tendons, no. We're talking fibers, so we're talking thin and fine inside the matrix. But collagen fibers are going to add firmness and tightness. And some of you ladies or men have heard of products that have collagen in them. And what's that supposed to do for you? Firm and tighten your skin. Right? No, Botox is different. That's a botulinum toxin, which destroys nerve function so you don't have wrinkles. I think I'd rather have wrinkles. Thank you very much.
What's that? They're doing like this new thing for people that have migraines. They're doing
Botox injections.
They go once she goes like once wrinkles, once afterwards.
Really?
That.
I don't know. I never heard of it. I'd have to look it up. And interesting. So anyway, collagen fibers, the thick ones, add strength. We also need stretchiness. See what I'm doing? I'm pulling my epithelium along my... So I need a little give there, right? So the other thing I'm going to find in connective tissue is elastic fibers. Some have more than others. Yes? Yes. And then the third one is something called reticular fibers. These are kind of like fine little spider webby fibers. They sort of act like a background for all the cells and things to sort of stick in that matrix. So when I think of connective tissue, I think of Jell-O with fruit in it. Yes? Kind of all floating around in there. Now, I'm not good at the whole Jell-O mold thing. Mine always come out a little squashy, right? Sometimes there's not a lot of water in it. What happens to the Jell-O mold? It gets a little tough, okay? So we're going to talk about the connective tissues that follow. And we're going to talk about the different amounts of these substances and how it actually changes the texture of that connective tissue so that it can perform different functions for the body. And that's where we'll pick up on Thursday. Adios.